Welcome everyone to my presentation on FATE, Feature-Agnostic Transformer-Based Encoder for Learning Generalized Embedding Spaces for Flow Cytometry Data. My name is Lisa Weijler and I'm from the Computer Vision Lab at TU Wien in Vienna, which is located in Austria. Recently, the trend in the deep learning world has gone more and more towards more generic architectures and training strategies, where a lot of research emerges around so-called foundation models. Basically models that are able to extract very generic features trained without a specific downstream task in mind. Often such generic models support data multimodality in order to extract strong general features, most commonly combining text and images, but the number of modalities is increasing. However, usually to extract those multimodality features, those approaches rely on unimodal encoders to process the different input data types and features, which are then combined using, for example, cross-attention. The input features in these unimodal encoders are fixed, however. They are the same for each data point, such as RGB values or text token embeddings of fixed length. In this work, we analyzed how we can make models more generic with respect to those input features, to support a varying amount and order of input features without using different encoders. And one use case where this is particularly important is the automated processing of flow cytometry data. Flow cytometry is a method to analyze blood or bone marrow samples on a single cell level. One flow cytometry sample is a set of feature measurement vectors, each corresponding to a single cell, also called event. Here each row is one feature vector and the columns are the features. The choice of the features measured and used for the analysis is highly task dependent and often varies even within one use case due to the different medical protocols, labs and routines. So often different samples have only a small proportion of overlapping features, but usually all sample-specific features are important for the task at hand. For automated cell detection, such as cancer cell detection, we need to process the samples in a consolidated way. But with the current architectures, this is not possible, and having different encoders per sample is intractable. One would need to either dismiss features or to take the union and impute missing values, which is why we designed FATE, an encoder that is able to process varying input features and learn a general embedding space. So how did we do it? In general, we exploit the characteristics of the transformer architecture, namely that it is equivalent to permutations of the input sequence and can handle different sequence lengths. We exploit those properties in two ways. First, to be able to process events with different numbers and ordering of features, and second, to be able to process samples with varying amount of events. We can separate our architecture in two parts. The feature encoder part, which corresponds to processing varying features and learns the relationship between them, and the set encoder part, which corresponds to processing varying amount of feature vectors and sets the events of one sample in relation to each other. This is a complete overview of the FATE encoder, so let me walk you through now. Each color depicts a different type of feature. On the bottom left, we have two input samples illustrated. As input to the model, we treat each feature vector of one input sample as a sequence of scalars. And so that the model knows what scalar corresponds to what type of feature, we employ feature encoding similar to positional encodings. The feature encoding is a learned vector that we concatenate to the scalars. Those are then processed with the feature encoder and further with the set encoder, resulting in a general embedding space with fixed length feature vector, regardless of the number and ordering of input features. Finally, cancer cells are detected with a simple multi-layer perceptron prediction head. We further propose a mask autoencoder pre-training strategy to learn the relationship between the varying features and create this common embedding space that then later is used for the downstream tasks such as cancer cell detection. The common masking strategy is to hide and reconstruct a full data instance that would be full feature vectors in our case. However, Since we want to learn the relationship between features, we propose to mask single feature values instead, random with a certain percentage per feature vector. Here, the full architecture of the unsupervised training strategy is depicted. The decoder has the same architecture as the encoder, but reversed. Since the input to the decoder is the general embedding of the sample, we need to give it the information of what features to reconstruct. We do this by simply also inserting the feature encoding of the features we want to reconstruct to the decoder and use cross-attention. Moving on to results. In our experiment, the goal was to detect cancer cells in pediatric acute myeloid leukemia patients. AML is a rare and highly heterogeneous disease with limited amount of data. Hence, utilizing available datasets for pre-training is an important matter. The target dataset consists of only 71 samples. There are, however, other AML datasets, either without cancer cells from screening cases or from different time points, and hence not comparable and useful as training set directly. Also, we use a dataset of different type of leukemia, acute lymphoblastic leukemia, with only 4 overlapping features with the target set. We compare it to the state-of-the-art method in this task, the set transformer. Since this architecture requires a static amount and ordering of features, we use the union over all appearing features and imputed missing ones with 0. While training from scratch yields similar results, when leveraging the additional data for supervised pre-training, we can see that the gap between the baseline and our proposed architecture is increasing. Interestingly, when adding the data from the different leukemia type, the performance of the set transformer is decreasing, while FATE can still leverage the additional data effectively. The highest results are achieved when using additionally our proposed masked autoencoder pre-training strategy, which results into a performance increase compared to training from scratch of approximately 60%. Here you see 2D projections of a target AML flow cytometry sample, where each point corresponds to the feature vector of one cell. The first rows correspond to the original feature space, the second row to the learned generalized embedding space. The different colors denote different cell types. Here we can see that similar cell types, cell clusters, adhere to each other also in the learned embedding space, showing that FATE is capable of learning meaningful embeddings. Summarizing our work presented today, we've introduced a novel architecture FATE and masking strategy that can process varying input feature sets and creates a meaningful general common embedding space. Further, we have shown that our architecture can successfully leverage the information of additional data sets, even with different leukemia types. And finally, using our architecture and masked autoencoder pre-training strategy, we yield a performance increase of approximately 60% compared to training from scratch.